Good afternoon, everybody. Welcome to Fascination of Plant Days 2015. So today, what we are going to talk about is that we're going to talk about the good life needs the good plants. That's the motion starts, and so we will talk about how we can utilize the plants in our daily life and in our future for food security and other things as well. So today, basically, I'm going to talk about the plant genome sequencing. And if you were to look at the history of the genome sequencing, you will see that it started from using Arabidopsis and continue all the way down to now we're sequencing wheat, which has a big genome. And while we're doing all these sequences, draft sequences, the cost of the genome sequencing is going down and down, so that we will have a lot of genome sequences of the plants we can utilize, manipulate the genes, edit the genes, and we can use in our life. So one of them is recently one of the model grass species that has been sequenced, the Brachypodium, and published recently. We were also involved in the International Brachypodium Initiative to sequence the Brachypodium, and recently in 2014, and this was a different approach, wheat genome has been sequenced as well, a chromosome-based approach, and now it's currently available. But the question that we need to ask is if we want to have the genome sequencing fast or cheap, what kind of methods we need to utilize, because there are a lot of platforms available currently, but the cost is going down. The question that we are asking is that how we can optimize the quality and cost ratio so that we can have a good quality of the product. So when we look at the human genome sequencing, it started really early in Bruce Alberts, president of National Academy of Science. He talked about sequencing of the human genome is going to help a lot to human and then medicine as well for the 21st century. When we change the three words here, the complete wheat genome sequencing will also help in terms of food security and then science too. So having those wheat genome sequencing, the good things that we need to have is that we need to have a germplasm and products in a short period of time so that we can utilize that genome sequencing